Hello, can you hear me? Hello, welcome. We'll get started in just a minute here. All right, it is 4 o'clock, which means it is time to start. My name is Hunter. I'm a post-bac student at Portland State University, working on pre-med requirements, and I was asked if I could help Principles of Biology students with reviewing content that will be helpful to you for exams. So if you have any questions, feel free to drop them in the chat. If my background music is at all distracting for you, I can turn that off, no problem. You might hear my puppy in the background. I have a little blue heeler Chihuahua mix who likes to check in here and there. Right now she is sleeping peacefully. Her name is Goose. You'll probably get to meet her. But with that, are there any questions before I get started? All right. Hello and welcome. The last thing I wanted to mention before we get rolling here is I understand starting next week you may be eligible for extra credit in your Principles of Biology courses. And we're setting up a Google form right now to help facilitate keeping track of who's viewed the live streams and worked on the worksheets. And I will theoretically be looking at the worksheets and grading those. So grading those for completion. That's all the notes that I have. Let's go ahead and get started. So today we're reviewing plant signaling, plant nutrition. What are we reviewing today? Plant nutrition and signaling. This is kind of a fun worksheet. I had a good time kind of previewing this one and going through it last term. So, a little bit of thought experiment first. Long-range manned space travel and manned space stations may someday become a reality. Before this can occur, however, we will have to develop sustainable methods of agriculture suitable for use in space. One of the key methods being investigated is hydroponics, growing plants in water supplemented with nutrients. You're assigned to a team working on the design of plant growth systems for use in a space station. First question. What types of plants would you choose to grow? Explain the reasoning behind your choices. This is really open-ended, so I see we have a couple people in here. If you have any ideas, feel free to throw those out here. There are many possible answers to this question, but the stuff that we need to keep in mind is what do the plants need, what do they produce, what other chemical needs are there, what other organismal needs are there going to be. So, for example, if we're looking at crop plants, how much of the crop plant produced can be consumed or otherwise used by humans or any other organisms. If anybody has seen the movie The Martian with, I'm going to blank on this particular celebrity's name. Came out in 2015. Oh, Matt Damon. Really fantastic example. Grew potatoes in, I think, his fellow astronauts' poop when he gets stranded on the space station on Mars. Really fun. So let's start entering some information here. That was an awful lot of talking. So oxygen and CO2 are, of course, are going to be really important considerations. Caloric needs, and then let's see here. Yeah, I'm just going to throw in here. Potatoes might be better than wheatgrass if we're trying to keep people nourished and alive. And in terms of the resources needed to grow those potatoes. Cool. All right. So then we're given this really dense data, which it looks like my app has decided to split a couple of times. So a couple different questions here. When we set up our growth system, part A, what nutrients would you need to add to the water? List what you would need and explain why each nutrient would be necessary. So we're looking at this table 36.1 in your biological sciences textbook. And it's a fairly complete list of all of the micronutrients and the macronutrients required for plant growth. And the table it also indicates the major functions of each of those nutrients. So you'll get a sense for what the biggest need is, but also how to know when your organisms are running low on specific nutrients. So right at the top of the list, obviously, we have oxygen, carbon, and hydrogen. Carbon's a really easy example. If you're short on carbon, it can't synthesize sugars, you're going to have slow growth and starvation of the plant. And on and on. Plants need nitrogen to make protein, without which they'll fail to thrive. You'll get, sorry, I forgot to scroll down here for you so you can see what I'm seeing. Chlorosis, that yellowing of the leaves. I actually just trimmed off some leaves on my prayer plant that were looking very sad and yellow at the tips. I should probably replant this, but I'm waiting for it to get a little bit warmer before I do that. So rather than go down the list and name what compounds we're going to need to, or what nutrients we're going to need to add to our water, I'm going to just let this list sort of exist by itself, and you can read through, and I do recommend starring next to the ones that you suspect will need to be added, and those are the ones that are going to be missing from whatever ecosystem you create on the space station. Part B, what atmosphere would you need to maintain? List what components you would need to maintain in the atmosphere and why each component would be necessary. So let's, big picture. We inhale oxygen, O2, that the plants have produced through photosynthesis. We exhale CO2, and that CO2 gets reincorporated into starches in plants. So let's keep it really simple there. Plants need CO2. And on and on. So again, I would review this list on your own and put a different shaped icon maybe next to those that are atmospheric. CO2 is a really easy one, but you might also be looking at, what else we got in here? Nitrogen is usually going to be taken up in the soil with the help of mycorrhiza. Yeah, I'm going to leave it at that. CO2 is a big one. Part C. You know what? I'm going to rewind a little bit. So the atmosphere in your space station would have to be similar to the atmosphere on Earth. I can't leave it with put CO2 in there. Your other organisms are all going to die, right? So this would be a requirement for both human life and successful plant growth. The plants are going to, again, require that carbon dioxide for photosynthesis and oxygen for cellular respiration. And if that oxygen to carbon dioxide ratio becomes too high, then C3 plants in particular would undergo photorespiration instead of photosynthesis, and this would reduce the plant growth and crop yield. So you'd obviously have to balance the number of plants versus animals and other carbon dioxide producers, maybe microbes that might be important to have on your station in order to control those oxygen to CO2 ratios. So, for example, if you grow legumes, symbionts associated with plant roots the rhizobium would be really, really important. Those will help fix nitrogen, which would reduce the need for additional nitrogen fertilizer. If you've ever heard of crop rotation, this was like a big innovation in early human agriculture, where we are alternating what crops you grow. Wheat for a season, and then or a couple of seasons, and then you grow legumes for a couple of seasons, because those mycorrhiza, that rhizobium, fixes nitrogen and restores the nitrogen in the soil which the plants need to make proteins for growth. So now we can move to part C. Which of the requirements in parts A and B could be recycled? Which of them could not be recycled? And explain. So your oxygen and your carbon dioxide are being recycled, right? Between the plants and the animals on the station. Water in the atmosphere is going to be recycled. You've got urination in the animals, photorespiration, things like that. Many of the minerals in the plants could be recycled by composting those inedible parts of the plants and animal feces. Those could, of course, then be converted into inorganic nutrients for the plants by the actions of various microbes. So I'm getting a little ahead of myself, but let's see if we can do some typing here. I'm doing this in rough list form here. Minerals. So we have chickens in the backyard and compost piles and plant matter, and we grow a lot of our own food here at my house. And I am starting to think that I should maybe refer to my home as our little space station, because we have a nice little self-contained ecosystem where we're producing all of this plant matter, which then goes to the chickens, which produce protein in the form of eggs. And then that the excess gets fed back to the chickens, which turns into poop, which turns into compost, which goes in the garden. So you're starting to see like how these systems already exist around you, right? This notion of the fantastical space station is not so far-fetched. These are systems that we interact with all of the time. All right. Microbes are very, very important in all of this. So let's move on to question 3. You're interested in growing soybeans that have a higher amino acid content than those varieties that are currently available. How would you do it? So how do you make amino acids? Okay, we've got a question in the chat. Thank you. Do rhizobium function with hydroponics? That is an interesting question that I don't know the answer to. There are many, many big question marks about hydroponics. But if we're going back to that earlier question, right, one of the advantages is how little surface area that you need to grow those plants, theoretically, right? But this feels like something worth Google searching. Hydroponics. I definitely misspelled that. The impact of rhizobial inoculation reduced nitrogen supply on... The first thing that came up was an NCBI study, which does not seem to mention hydroponics. So I'm going to put hydroponics in quotation marks and see what we get. Testing rhizobial inoculation of common bean in hydroponics enables accurate quantification of biological nitrogen fixation. So it looks like it is being studied, which is kind of cool. Seems like something that would be worth looking into a little bit more if you were interested in that. Yeah. Awesome. So actually, it kind of seems like you're right on the money here. So the only way to increase your, with regard to this particular question, the only way to increase your amino acid yield or the nutritional composition of any fruit or vegetable is by selection and breeding of plants with the desired characteristics or genetically modifying a variety for higher amino acid content. And of course, you're also going to have to make sure that those nutrients are available for those plants in the first place. So if we're assuming a perfect system where all of the nutrient uptake is optimal, then the only way that you're going to get soybeans that produce more amino acids is by selectively breeding those over many generations. So you may also fertilize with nitrogen or inoculate with rhizobia, as you were alluding to. But that's not going to change the nutritional composition of the fruit, right? So, cool. Again, all of these questions are pretty open-ended, so whatever you come up with, as long as we're kind of on the right path conceptually, I think it's going to be, should be your focus here. There's no one right answer. Where were we? So questions 4 through 5. Pine seedlings grow in sterile potting soil much slower than seedlings grown in soil from the areas where the seeds were collected. Which of the following is a reasonable explanation? And explain your answer. A, what are we looking at? Oh, I see. The normal symbiotic fungi are not present in the sterilized soil. Is this a reasonable explanation? Would anyone in the chat like to chime in? Would a lack of the normal symbiotic fungi cause the seedlings to grow slower in the sterile soil? With confidence, yes. I would say absolutely. This is absolutely a reasonable explanation for the slowed growth because plants that have symbiotic relationships with mycorrhiza are better able to take up nutrients, essential nutrients, especially nitrogen and phosphorus, from those soils. Without the ability to take up large quantities of nitrogen and phosphorus, plants would grow much more slowly. So yes, this is reasonable. Cool, didn't misspell it. Is the tapping of the keyboard driving anybody crazy? Should I get rid of it? Because I can mute while I'm doing that. Any feedback is helpful here, guys, not just about the keyboard. Moving on. Question 5. Is water uptake faster when mycorrhiza are present? Would that explain slower growth in the sterile soil? I would say this might also be a reasonable explanation. Your textbook may not indicate that mycorrhizal fungi contribute to this in a major way, to increase water uptake. However, and this is just a cool side note, some researchers have found that certain mycorrhiza do increase both water and nutrient uptake. So yes, this is plausible. Cool. All right, on to a new set of questions. My scroll wheel is out of control, y'all. I promise I will have that fixed by the next broadcast. You observe the results shown in the graph below by measuring plant growth in 2 different soils, soil A and soil B. Assuming a difference between the soils is the presence of mycorrhiza in one soil and its absence in the other, which soil has the mycorrhiza, and how do you know? Any thoughts? So we've got our 2 growth charts here. Plant growth on the y-axis, time in weeks on the x-axis. And we have a divergence here between the 2 growth charts with, looks like soil A plateauing around 10 grams. and soil B continuing up the chart. Seems like soil B has the mycorrhiza. I would agree. Plant growth in soil B is much better than in soil A due to the presence of fungi that help plants obtain nutrition. In part B, propose an explanation for why the plants in soils A and B grow at nearly the same rate for the first few weeks. Any thoughts? So we've decided that soil B has the mycorrhiza, but soil A does not. That didn't seem to matter until after week 8. Why might that be? Nutrition that's in the endosperm evens things out for the beginning. Yeah, absolutely. Most plants growing from seeds have plenty of nutrition for the first couple weeks to sort of kickstart that process. And depending on what signaling process the seed uses to initiate that growth phase, I'll come back to that. So yes, one explanation is that seedlings have a lot of nutrition stored in the endosperm of the seed and therefore do not need a lot of external nutrition at first. Another explanation is that when a seed is first planted, there could be ample nutrition in the soil directly surrounding the first roots that form, and so those can be easily obtained by the plant. After those nutrients are depleted and the plant gets larger and needs additional nutrients, you might observe the effects of nutrient depletion if the mycorrhiza are not present. Same reason you got to repot here and there. Okay, so yes. Cool. So next we're going to look at gravity and light affecting plant growth responses. Question 1. One of the problems associated with growing plants in space is the lack of gravity. How does gravity affect the normal growth of plants' roots, stems, and other parts? Explain the mechanisms involved. Does anyone know how would sunlight be a factor with the growth as well, or is it only the soil? Sunlight's absolutely going to be a factor, and that's what this section of the worksheet is all about. We're just kind of kicking things off with gravity because it's also important. Has anyone here taken anatomy and physiology or animal biology yet? It's like the different ways that plants determine up from down are multifaceted and different from how animals determine up from down. So And one of those is going to be sunlight, and we'll get to that. So under the influence of gravity, there's this chemical auxin, AUXIN. I'm going to go ahead and type that in. Auxin accumulates on the lower side of roots and stems. Higher auxin concentration stimulates cell elongation in the stem, but it inhibits cell elongation at the root. So in the shoot region, well, okay, let's go and write that down. High auxin equals cell elongation in stem, but not the root. In the shoot region, shoot, above ground, a concentration of auxin between 10 to the negative eighth and 10 to the negative 4 molar, concentration, not quantity, stimulates cell elongation. According to what's called the acid growth hypothesis, auxin concentrations in this range stimulate proton pumps, which lower the pH in the cell wall. And this activates enzymes that break cross-links between cellulose molecules and allow the cells to elongate. So, ten to the negative 8 and whoop. I'm going to go ahead and put here, I'm going to bold this if I can, because I remember this being important for exams. I cannot confirm that directly, but there's a key word here, and that is, I would spend a little time learning more about acid growth hypothesis. Right, so under the influence of gravity, auxin accumulates at the bottom of those shoots and roots. And so because of the different responses that roots have versus shoots, the plant can differentiate where it should direct resources for growth. How would a lack of gravity affect normal plant growth then? So somebody hinted earlier at the role of sunlight. Lack of gravity would affect it with its primary growth. What do you mean by primary? Primary as in early growth, or primary as in largest components? Of the plant. I'm going to go ahead and answer the question for you. Early growth. You are exactly right. So later, as plants get larger, sunlight is going to have a lot more to do with, evapotranspiration. This was covered, I think, last week, in terms of pulling water and nutrients up through that evaporative process, creating negative pressure in the vasculature of the plant. Especially early in the plant, like seeds are buried underground. They don't know which way the sun is. So this gravitropic, that orientation via gravity, is entirely how plants know which way to go in the absence of light. So Let me make sure I'm... So in the absence of gravity, auxin concentrations in the root and shoot would not vary. And growth of the shoot away from gravity and growth of the root towards gravity would not occur. Amyloplasts. I'm not... Give me 1 second. I'll just say directionless growth or stunted growth. Let me make sure I hit on everything. Cool. I'm going to do a quick vocab check because I'm a little rusty. Colorless plant plastid that forms and stores starch. Found in many tissues, particularly in storage tissues. I don't know. So the gravitropism, to review, the gravitropism, orientation with relation to gravity, is more to do with those auxin concentrations because it changes with respect to gravity. Higher concentrations, lower in the plant. In the absence of gravity, concentration of auxin throughout the plant would be the same, and so the plant won't know which way is up, especially in that early phase of growth when there are no shoot systems above ground for it to orient with relation to light or phototropism. So, propose mechanisms to overcome the problems associated with the lack of gravity. We kind of already touched on this, right? Plant orientation is also affected by light, and the response to light can help counteract the lack of gravity. Always assumed roots grew at a similar rate to shoots, but is that not the case then? Yeah, based on that acid, based on I don't want to tell you the wrong thing here, so I am checking my work as I go here. According to the acid growth hypothesis, auxin concentrations stimulate proton pumps, which lower the pH in cell walls, which activates enzymes that break cross-links between the cellulose molecules. But that wasn't your question. Your question was, do the roots grow at similar rates to the shoots? So, higher auxin concentrations stimulate cell elongation in the stem, but inhibit cell elongation in the roots. My recollection is that root elongation is triggered by, or stimulated by, the presence of other compounds and other stimuli in the soil. And so, for example, that's how it knows how to grow around obstacles rather than just stopping. But auxin specifically seems to be about shoot growth, so stems, in this particular focus. Yeah. So let's propose some mechanisms for overcoming this. Light was one. What else? Who's familiar with hydroponics? What features of hydroponics could help us overcome this? Possibly magnets. Interesting. Okay. I know some of the essential nutrients required for plant growth are, well, maybe, but I don't think that's our first fix. Isolate auxin and give it to the plant. Interesting. So, like, injecting it at specific places where we want it to grow. I'm going to go ahead and write that down. Cool. That is not on my list of proposed solutions, but like this is how innovation happens, right? Brainstorm. We're doing it. We're going to the moon. What else? One of the examples that I have here is when you grow plants hydroponically, you're very often using these dense mesh mats to create a substrate for the roots to grow in. Electromagnetic radiation stimulate photoelectrons. In the absence of light or in the absence of gravity? I'm not... You're having a big brain moment and I'm having a hard time keeping up. I don't know. I don't know. First we gotta overcome like certain limitations just in terms of the roots knowing where to go. Part of that can be overcome just by creating somewhere for them to go. So those dense mesh mats could be used to prevent roots from growing above the water, for example. And water coheres to itself pretty well, so it'll just stay where it needs to be. Especially if you give it something hydroponic or hydrophilic to stick to. So dense mesh mats for hydroponic. Hydroponic, not hydrophonic. We're not making music with the water, we're making plants. Trying to. Space plants, specifically. Yeah, let's do that. So another problem with growing plants in space relates to a plant's light requirements and phototropic responses versus photoperiods required for the plant to flower and produce fruit. How do phototropism and photoperiodism differ? And even without having my cheat sheet here, I'm kind of thinking about the breakdown of those words. So photo, light, tropism, location. What is the meaning of tropism? Orientation. The turning of all or part of an organism in a particular direction in response to external stimulus. Okay, so we'll go with orientation. Periodism, periods, periodicity has to do with when the growth happens. You're right on, exactly. So phototropism has to do with, exactly. Phototropism has to do with direction of growth, orientation with respect to light. Photoperiodism has to do with when the growth happens, or when the light is present. Maybe put the plants in an airtight container and have it grow towards water and light. Airtight container. Grow towards water and light. Talk me through the airtight part while I write down a few things. Phototropism, plants orientation lives. Seasonal changes in light. Yeah. Yep. Where is my burp? Come on now. Oh, do you not have the worksheet? Okay. Well, you guys are obviously not being graded on this. I am so sorry. Yes, let me create a link. And I'll drop it in the chat here. Yeah. Copy link. Anyone with the link can view. My recommendation would be to... This is a Word document. Yes. Open this in Microsoft Word and then edit the Word doc. Or if you're going to, or you can print it off and write on it. I'm opening this in OneNote because that lets me do fun things like, let me see if it'll let me. Yeah, like that. Do I have a worksheet for the previous live streams as well? Yes. I'm working on getting email lists for all of the Principles of Biology classes, and I'm also in communication with Dr. Reddy about this. I will make sure that you all have access to these worksheets one way or another, whether that's by shooting an email blast with a share link for this folder, or putting it in Canvas or something like that. I will make sure that you guys have access to these before next week's stream on Wednesday at 4. Cool. Thank you for checking in on that. I didn't realize you guys didn't have access yet. If anyone else knows where to find those, do chime in. I don't have great visibility on that back end of the process. Yeah, no problem. I will say, one of the reasons I'm doing this is because I had a really fantastic experience during my Principles of Biology class with attending these live streams. Of course, how everyone was handling code was a little bit different when I started school too, so a lot more of it was remote or hybrid learning. And I really appreciated kind of being able to sit at this desk and watch these streams and work through things. And also, your learning assistants and workshop leaders are really, really fantastic resources in terms of figuring out study strategies that work for you. I did not have really solid study habits going into school this time. I have a bachelor's degree from a different university from a past life, and coming into it this time, I was determined to ask for a little more help when I needed it, And so I want to pay it forward. If you guys have any questions about that, email me. I'm going to drop that in the chat here as well. It is hlea@pdx.edu. Email me anytime. I'll help as much as I can. If we have time, I can also dedicate a broadcast to that. But one of the ways that you can study well for your Principles of Biology class is hopefully attending these broadcasts and reviewing these concepts together. Postbac student myself, and I'm learning as I go along, learning how to be a better student, especially with biology. Yes. Right off the top of my head, we're going to come back to that because I will not get through the material that we have left in the next 13 minutes if I do not. I have a little bit of time today. I can stay late on the broadcast and we can talk. So, okay. So we talked about phototropism and photoperiodism and how they differ. What light characteristics would you use to maximize plant growth per unit time? This is our review of photosynthesis. Blue light to stimulate the chloroplasts. Interesting. Okay, why blue light? You're on the right track as far as thinking of this in terms of different wavelengths of light, different colors. Blue light opens the stomata guard cells. So is the idea then you want the stomata to be open? So the stomata being open facilitates gas diffusion into and out of the plant, right? And a few other trace nutrients. But if you're, and water in particular. So having your stomata open all of the time could also dehydrate your plant, which could be bad. And I think that's triggered by, in humans, it's sodium potassium pumps. In plants, it is hydrogen, I believe. Anyways, I'm sure we will get to that at some point. So in photosynthesis, your chlorophyll molecules in photosystems 1 and 2 are different. Remember, photosystem 2 comes first. And respond to or absorb light in the red and blue wavelengths of the visible spectrum. So you're right on one count. And as a result, if nothing else is limiting, more red and blue light available would result in faster photosynthetic rate per unit time. So chlorophyll molecules in photosystems 1 and 2 respond to or absorb light in the red and blue wavelengths of the visible spectrum. As a result, if nothing else is limiting, the more red and blue light available, the faster the photosynthetic rate per unit time. It's both, right? So the different chlorophyll molecules in photosystem 1 and 2 allow plants to capture those different wavelengths of light because you don't have the full spectrum of light available all of the time. Carotenoids in particular are those, well, they're orange in color, right? And they're in higher concentration as the plant is winding down for winter. And so your photosynthetic output is lower, but those carotenoids are more efficient at capturing that lower angle sunlight, which tends to be less blue, I think. I might be reaching a little bit, and this isn't on my cheat sheet, but that's, I feel pretty confident in that. And that also kind of reaches back to that periodicity aspect. So going back to that previous question, you might want to change the amount of red versus blue light throughout your year on the space station to prompt those different seasonal growth patterns in your plants for flowering and fruiting. And we're on to C. So what kind of physical environment would you need to maintain appropriate phototropic responses among plants? What kind of physical environment would you need to maintain appropriate phototropic responses among plants? I don't know if you can hear my dog yawning in the background. What do you think? How do we set up our lights? I'm going to go with sun's overhead, lights should be overhead. Set up banks of overhead lights. Yeah, I think a daylight and a night cycle. What else? Let's go back to our hydroponic systems. Any unique needs there? Hydroponic systems require supports for seedlings and supports for stems and roots below grow mats. I have this image in my head of just vines kind of willy-nilly swaying with the movement of the spaceship as gravity gravity shifts with acceleration. Maybe not. All right. And finally, we're at the end of our worksheet and coming up on the end of our hour, so that worked out really nicely. What design modifications would you need to make to support plants with different photo periods? For example, long day versus short day plants. I don't know. I say let's isolate them. Different kinds of plants with different periodicities get different chambers. Does long day versus short day influence anything other than initial germination? Definitely. Yes, absolutely. Yep, you're both on the right track. So if different plant species had different photoperiods, need to be, grown in separate chambers. You could also have separate timer controls. To regulate the hours of light in each chamber. Per day. Cool. Yes, totally. You would need certain rooms that were dark for those who can't handle too much light all the time. So think about the different time zones at the various latitudes on our planet. And it's tilted on its axis, right? So if the northern hemisphere is tilted towards the sun, that's its summer. We're currently in winter in the northern hemisphere, so maybe that's a more appropriate example. But our days are shorter, right? Not all plants can handle uninterrupted sunlight all of the time. And this is definitely in the literature where resources get pushed to different processes within the plant at different times of day, right? So when it's hot out, you've got sun overhead, plenty of moisture, evapotranspiration's happening, pulling resources from the soil up the plant through its vasculature up into those leaves. So there's a lot of movement happening. There's a lot of photorespiration happening. Photorespiration? Hang on. No, you have photosynthesis happening. It's the opposite. At night, when there's no sun available, C3 plants in particular undergo photorespiration instead of photosynthesis. So they're closing those pores. Now I'm getting onto CAM4 plants, which you guys might not be looking at yet. Plants undergo different metabolic processes at different times of day, triggered by changes in ambient light, humidity, nutrient availability. How do evergreen plants survive in different lights and seasons like rosemary, lavender, and Mexican orange plants? These are all spectacular examples. So think about your pine tree and the way that those green, the photosynthetic parts of the plant are arranged, right? Yes, they're photosynthetic, but they're not optimized for it from an evolutionary perspective, right? In last week, we kind of talked about how the shape of plants informs their function and how you have really big surface area, flat, thin leaves to maximize surface area for photosynthetic output. That's going to result in a lot more it's a more efficient use of sunlight. So while your conifers are less efficient from a photosynthesis perspective, that also means that they need to be more efficient in other regards, and one of those ways is storing energy. If that makes sense. I am not a plant biologist, so I'm not going to speak a whole lot to that. I personally am much more interested in human physiology, but we interact with plants for lots of things. Xylem. This is an exam question. One of the most useful products from plants to humans in terms of building shelter is xylem. Wood products are all xylem. All right. It is 5 p.m. and that is technically the end of our broadcast. If you are here for these worksheets and if you are watching this as a recording later on, you can go ahead and call it a day. We're not going to be reviewing this material anymore. Boy, now you're going to make me eat my words here. Wood is secondary xylem. Yeah. You are so welcome. No, it's totally okay. I also am reviewing this material, having only learned it a year ago, really. The rest of what I have is sort of, what would you call it? I'm going to get rid of that screen because we don't really need it anymore. The rest of this picked up in my life, and it's not part of a formal scientific education. This is my first time studying STEM. The only science class I took in college the first time around was a historical geology class, which was awesome, especially being in Colorado, but this is very different, and I'm having a really great time with it. So this is as much review for me as it is for you guys. I just kind of have a little more context because I'm a little further in my studies here. So thank you for being here for the live broadcast. I am going to make this video available for people to watch after the fact so they can work on the worksheet on their own. And I believe this is also going to get uploaded to the Learning Center's YouTube channel eventually, once I get instructions on how to do that. So that concludes the broadcast. I'm happy to stick around for another, we're going to say, I'll stick around till 5:15 if you guys want to talk about study habits and study strategies, especially as post-bac students. And I don't know if anyone else here is a career changer, but yeah, we can talk about that. So fire away. I will hang out for another 13 minutes. Study strategies and my first degree. I studied French. I was a foreign language major, ultimately majored in French, but I studied Russian and Spanish as well. And I could still get along in French, but probably not the others. Also post-bac, also first time in STEM. Awesome. Si, c'est vrai. J'ai fait mes études à Tours. C'est une petite ville au sud-ouest de Paris. Et puis Paris aussi. So I did study in France for a while in a little place called Tours, which is southwest of France. About a 2-hour train ride. Yeah. So did you also study foreign languages, or are you ESL, or is French your native language? Thank you. Oh, this is wild. Cool. Haven't needed my French in a hot minute. As far as study strategies, I didn't have to work very hard to get through high school, and so I didn't have any skills built up the first time in college. Your degree was in French and applied linguistics. Mine was in, language and literature. So I did a focus in post-revolutionary absurdist literature. I wrote my thesis on Albert Camus' The Stranger and cyclic moral development. It's a whole thing. Film and literature. Nice. Excellent. Cool. Here we are in STEM, just becoming really well-rounded humans. I love it. I love it. Study strategies. Because I'm on pre-med track, I was alerted to the existence of Anki really early on. That's A-N-K-I. Let me just pull up the website for you. I think it's ankiweb.net. Yeah. Highly recommend it. There's a free app for Android users. The iPhone one is $20. It's totally worth it. It's free. It's a spaced repetition learning platform, and it's very powerful, especially if you have any programming chops. I have no programming skills, and I was able to figure it out. So what Anki is, it's not a learning tool, it is a study tool. So it's for reviewing stuff that you already understand. It's also good for vocabulary. I found it extremely helpful in combination with, there's a plugin for it. There are tons of YouTube tutorials on how to set up Anki for success, especially for pre-health track. The power of Anki is all of the pre-built decks that already exist, and the ability to make tailored decks specific to your class. So if you can find decks specific to the textbook that you're currently using, that will be extremely helpful. And if they're tagged well, then you just unsuspend, it's called, the tags for the subjects that you're currently studying. So you review the content for the chapter, unsuspend those cards, review them, learn them, and then you basically tell the computer how quickly you were able to recall that information. You score it from 1 to 4. 1 means you need to review it sooner. 4 means you can push it out a ways. and the software automatically kicks those spaces those flashcards out further and further. And it's based on really, really fantastic research in terms of the periodicity. There's that vocab word again, of the optimal periodicity for active recall of complex information. So again, it's not a learning tool. Don't use it that way. It's a review tool. It's a study tool. I still review the cards from my Principles of Biology class. They are still in my rotation, and I do them every day. That was the first really, really big breakthrough for me. The second was organizing my notes really, really well. And I might be able to share my screen here again. Hang on. Let me just make sure I'm going to show you guys like my Google Calendar or anything. So, okay. Yeah, I'm going to show you my organic chemistry notes because they're awesome. Share screen. Just downloaded Anki today. Congratulations. I may even be able to upload my decks from Principles of Biology and make those available to you guys. I will look into that. So what you guys are seeing right now is my OneNotes app, and it has every lecture, every chapter, every study session, all of my practice problems. And last term I arranged them all by chapter, and these are all arranged alphabetically so they appear in some kind of logical order. What I have changed to, because I like my brain recalls things chronologically a lot better than it does topically. It's like, when did we learn that? Nowadays I'm doing everything by date. And so, with a few exceptions, my notes are arranged by date first, then by chapter, and then activity. Was it lecture? Was it worksheet? I'm doing a rolling doc for my quizzes where I work through them and then write out my rationale and then validate, did I get that right? So that's three really big techniques that kind of are working well for me right now. Is the spaced repetition with Anki for vocab, for reminders, for I should just open Anki. If you don't know already, journals, what's nice, and I think it was developed by a guy to help people with ADHD plan better, basically, because it's a really flexible system. But the power of the system is it allows you to keep track of where information is within the journal. And I have mine set up using kind of those same principles, and it has worked very well. The power of the system is the flexibility. Anki works for the same reasons. Okay, so it's taking things a minute to boot. I'm asking a lot of my little tablet right now. It's trying so hard. Okay, so I have Word. I see you. Okay, so I have a deck for all my post-bac courses. And then sub-decks, these are called, for each class. And then I will have decks inside of that for each subject. You can see these are not contiguous. I didn't use them a whole lot, and there's especially in chemistry, like there's very few cards in them. They're just for like specific formulas or numbers that I needed to memorize. And I don't have a ton of rotation right now, but what I can show you is what this looks like. Study now. So this is for my OChem class. These are SN1 reactions, and you can see this is a closed deletion, so substrate effect, I'm supposed to rank this in terms of stability from methyl groups to a third degree carbon. Guys, thank you so much for hanging out. Appreciate you. And then, so SN1 reactions, substrate effect. I believe that methyl groups are the least stable, and I am correct. Methyl groups are less stable than primary, are less stable than secondary, are less stable than third degree, tertiary. So I tell it that I got that right by hitting the space bar. What in the world? Yes. And then it takes me to the next one. That's a image occlusion. So those are 3 big ones. The last one is just treat it like a job. I know a lot of folks in college are a lot younger than I am. I'm in my 30s. And it is perfectly okay. It is optimal, in fact, to have boundaries around your time and only work on school during school time and to have a life outside of that. Because losing sight of why you're here will burn you out very, very quickly. So I'm going to use that as an opportunity. Yeah, I'm going to use this as an opportunity to show y'all my the love of my life. Come here, Goose. Yeah. Are you the longest noodle? Burp. This is Goose. She reminds me to drink water and take naps. Hi, baby. Yeah. And she's been my study buddy since going back to school. Yeah. So. Dog. Yes. This is my baby. Yeah. You're gonna live forever, aren't you? So. Any last questions? I know there's only one of you left here, but if any of this is helpful for you, it'll probably be helpful for somebody else. Hi, Goose. They love you. Yeah. You're a star now. Yeah. You're smelly. You need a bath. Probably been playing in the chicken coop where you're not supposed to be. All right. It's 5:15. I'm gonna get going. Cool. Awesome. You are super welcome. Before I let you go, because this might be helpful to someone else, definitely go through your syllabus and prioritize the things that you absolutely have to get done and do those first. All the extra credit stuff like this should be secondary. Don't worry about getting that credit if you're not nailing it in the other high point value stuff. It is a game and we got to play it a little bit. So, cool. All right. Good night, everybody.